In this next session, it's our last of the afternoon, we're going to hear from two researchers who have submitted abstracts for a short data blitz presentation. So we had many poster submissions this year, so really excited to share these two with you. First, we'll hear from Mason Wilkinson. He's a PhD candidate at Vanderbilt University. And then Anne Sophie Leah. She's a senior lecturer and hospital practitioner at the University of Le Mans, France. So Mason, take it away.
Where do I advance to the next slide here? This little guy? Okay. Hi, everyone. This is going to be really quick. So today I'm going to talk to you about a small molecule high throughput screen I set up in rat Schwann cells and more focus on some of the cooler results I got down towards the end of that screen. And you can come talk to me at my poster later if you want to hear more of the nitty-gritty details. So first and foremost, it's been belabored at this point, but the majority of the mutations that cause Charcot-Marie-Tooth disease are associated with the PMP22 gene, predominantly the duplication or deletion of the gene that causes over or under expression, respectively. Now, by and large, the therapeutic strategies proposed for these two forms of the disease have been focusing on looking for ways that we can rectify wild type levels of expression of the PMP22 gene, bring it up for HNPP, bring it down for CMT1A. And in the Sanders lab, we've been investigating a lot of different ways that we might be able to do so using a small molecule that either alters the expression of PMP22 outright or brings up the trafficking efficiency to the plasma membrane in the case of point mutant disease PMP22. So to that end, in collaboration with Bruce Carter's lab, also at Vanderbilt University, I developed a rat Schwann cell line using a lentiviral transduction approach that expresses the locus you see up here at the top of the slide. And this is a genomically integrated human PMP22 gene with a MIC tag in its extracellular domain, and that is then flanked by a CMV promoter with a tetracycline inducible operator. So the idea is I can add a tetracycline to the Schwann cell culture media, and as we increase the concentration, you see here in this Western blot, doxycycline goes up, PMP22 expression also goes up, and the quantification is shown there on the right. My collaborator, Catherine Stefanski, who's a postdoc in the lab that's also giving a poster later, by the way, go check her out, did some preliminary immunofluorescence with these cells. So you're seeing that here in the top left. And as we increase the amount of doxycycline in the cell culture media, we see the red signal, PMP22, correspondingly goes up. But also, the number of intracellularly retained PMP22 foci increases as a function of total expression level. Extrapolating off of that is some flow cytometry data I did, showing here on the x-axis is the amount of doxycycline present in the cell culture media, and the black trace is then total MIC-PMP22 expression, while the red trace is the fraction of the MIC-PMP22 molecules that reach the plasma membrane. So we can see at really high levels of expression, we're essentially lowering the total fraction of PMP22 that reaches the plasma membrane. So to identify some small molecules that might be able to alter the total levels of PMP22 expressed by cells, either increase or decrease, I developed a strategy whereby I can seed cells into 384 well microscopy plates, simultaneously induce PMP22 expression at whichever level I want, and incubate that in the presence of screening library compounds for 24 hours, after which we can take a look at total immunofluorescence by microscopy. And what I'm showing here is just a few example images from a single treatment well within this high throughput screen. So the red signals PMP22. We counterstain for nuclei and the plasma membrane using DAPI and WGA respectively. And our microscope's inbuilt software can mark the location of individual cells within that image, and we can quantify cell by cell the amount of PMP22 being expressed, which we then extrapolate out to a population average, and we can compare different treatments. So very quickly running through the whole screening process, we screened just over 22,000 different molecules, rescreened to figure out what seems to give a consistent result. And we went through several levels of counter screening to evaluate compounds that are either toxic or things that appear to be affecting transcription from the exogenous promoter. And at the end of the day, we're down to 2 to 3 compounds that we're really interested in, 2 of which I'll tell you about here in a moment. So this is some example data from later in the screen. And what we have, let's go back. What we have on the x-axis is just a bunch of different compounds that we order for rescreening, the total amount of MIC-PMP22 at the cell surface relative to our untreated vehicle control cells is shown on the y-axis. So anything above the 100% line is something that appeared to be reliably increasing expression, while anything below that line seems to decrease PMP22 expression. Our top three compounds we're most interested in have very reasonable potency for this stage of the screen. Our 3 favorites have EC50 values of around 10 micromolar or less, with one compound, treatment 11, nearly increasing total PMP22 levels by around 75%, while saturating concentrations of the other 2 compounds, 14 and 25, almost entirely abrogate PMP22 expression in these cultured cells. Furthermore, my collaborator, Jake Hermanson, also at Vanderbilt, did some preliminary proteomics work. This is just a basic mud pit experiment looking at total proteome changes within Schwann cells treated with these different compounds. And on the left, we've got k-means clustering of different gene ontology families that are up or down regulated relative to the uninduced, untreated cells. And we're seeing different enrichment patterns for all 3 of our top 3 favorite hit compounds. And this may suggest differences in mechanism, but we're still looking into this. Also, interestingly, is that one treatment 14, does not appear to have any significantly altered gene ontology classes relative to an untreated control. So this could represent a high degree of specificity, but the jury's still out on that. Now, hit 11, that compound I showed you that increases PMP22 expression substantially, does appear to have pretty drastic alterations to the, in particular, glycoforms of PMP22 that are enriched during expression. So we can see in the untreated controls, we get all of our characteristic glycoforms, non-glycosylated, core, and complex. Treating with a glycosylation inhibitor, NGI1, enriches purely the non-glycosylated band, and hit 11 enriches in our core glycosylated band. Almost no complex glycosylation is observed in this treatment, and I expect that's probably bad. Nonetheless, this could be useful for teaching us something about how and why PMP22 traffics within the cell and how the glycan is involved in that process. What I'm most excited about, though, is hit 14. So this is another PMP22 Western blot in the presence of different inhibitors of either the proteasome or the autophagy pathway. And so You can see different glycoform enrichments as we treat with either the proteasome or autophagy inhibitors, bortezomib and chloroquine, and basically no protein whatsoever when we treat with this hit that reduces PMP22 expression. However, co-treatment with that hit and bortezomib, inhibition of the proteasome, appears to bring a substantial amount of the protein back. So it's possible that this protein or this treatment is somehow biasing PMP22 toward proteasomal degradation. And what I'll leave you off on is what we're currently working on. So what you see down here, these are DRG explant cultures. This is an iPhone picture taken of a computer screen. We are still in the early stages in optimizing this, but DRGs treated with this compound seem to be able to reliably form myelin, as shown by the MBP staining here. And beyond that, we're working on establishing mechanisms of action and trying to get the potency of these compounds down with SAR studies. So thank you very much for your time. I'd like to thank everyone in my lab and all my collaborators.